Okay, first thing to start off with, nobody asked me as we were going through the test last time about question 32. Just breezed right by it. Nobody said a thing, and I was surprised when I looked later and thought, oh, nobody asked me about that one. Which is good that I'm going back to it, however, because a lot of people had problems with question 32. Thankfully, it was only worth 4 points. Okay. Question 32, briefly explain genotype by environment interaction and provide an example indicating this phenomenon. Now, several people said it's when the environment influences the genotype. That is not what this is. Well, actually, just saying that the environment changes genotypic frequencies through time, that's what evolution is. Okay, is natural selection doing that. That is not what this is. This is the environment interacting with genes that you have in your body now. It is when the environmental conditions either turn on or turn off the expression of genes as they exist now in your body. This gives you what is called phenotypic. Anyone? What do we call it? Gives you phenotypic plasticity, meaning you're actually able to express more than one phenotype with one gene, depending on whether or not that gene is turned on or turned off. And it is the environment that can turn it on or off. What was the example I gave you? Hello? And it, not the bones. The example I gave you was the fact that when you expose yourself to excessive sunlight, ultraviolet radiation, you produce more melanin. Is that because you suddenly got a new melanin producing gene? No, you just stimulated the genes that actually produce the enzymes for making melanin to produce more of those enzymes for making melanin. Yes, yes. So it is the environment influencing the actual expression, which I think is the word that I probably wrote down in most cases, the expression of genes. Okay? Not changing allelic frequencies through time, that is just evolution. Okay? It is now in this organism, the environmental conditions change the expression of the genes. Yep. Yep. Please say something. You guys seem a bit loggish, this, and by that I mean kind of like lumps on a log. So
I'm here, I'm noting.
Okay. All right. Yes, we good? Okay. All right. So I had forgotten to go over that question. I figured I needed to go over that. Does anybody, after having looked at the test, need me to go over anything else on that test anymore? Like I said, I went over all the written part. I didn't go over all the Scantron part. Okay. Again, I'll say what I said before. Go over them. Understand why the right answers are the right answers. These are extremely useful tools for studying for the final, which is comprehensive. Okay. It's not that I'm going to ask the same questions, but it's the same material. So if you know why the right answers are the right answers, you should do very well on the final. Yes. Yes. Will some of the questions be exactly the same? Probably. Okay. But don't memorize the questions, understand the answers. Okay. All right. We had just finished our discussion last Wednesday about Hardy-Weinberg. Okay. And the assumptions of Hardy-Weinberg. And why Hardy-Weinberg is even a useful thing when discussing evolution. Why is Hardy-Weinberg a useful thing when discussing evolution? Because Hardy-Weinberg is the blank for evolution. Not well.
I get what you're saying.
It is kind of the proof.
Showing that evolution
Because, right. And it's the proof because you can disprove Hardy-Weinberg equilibrium because Hardy-Weinberg is the null hypothesis for evolution. Okay? That's why Hardy-Weinberg is a useful thing, is because Hardy-Weinberg carries with it all these assumptions, and all the assumptions are all about genetic frequencies not changing through time. But the point is that the assumptions of Hardy-Weinberg are almost never met, which means change is taking place, which means evolution is occurring. That was basically the last statement I had made last Wednesday. Yes? Yes? Yep. Okay. All right. So what we do in the rest of this chapter and in the next chapter is look at the 5 assumptions and talk about them. Because the 5 assumptions were no selection, no mutation, no drift, no gene flow, no, and then random mating. And we're going to look at all of those. And with random mating, we're going to talk about one specific thing because we're going to come back to sexual selection later because it deserves its own discussion. Very interesting stuff going on. Okay. All right. So basically, the rest of this chapter and the next chapter are talking about those assumptions of Hardy-Weinberg. So we're going to start by talking about the main one that I told you your brain should switch to when we talk about evolution, which is selection. That if you see differences between populations, the first thing that should come to your mind is that there's some selective differences between those 2 populations. It's not always the case, but that's the first thing that should come to mind. Okay? All right. So what's selection? Just make sure. That's where we're going to start. What's selection? Say that. Survival of, thank you. Okay? Survival of the fittest. Because that's what we ended with when we talked about the 4 postulates of natural selection, survival of the fittest. And what does that mean? What is biological fitness? Okay, that's one part of it. Surviving to reproductive age, it's got 2 components, surviving to reproductive age, and once there, actually passing your genes on, actually reproducing. Because your biological fitness is you passing your genes on to future generations. That's what biological fitness is. Yes? A little bit of review, never hurt anybody. Okay? All right? So, and of course, the point is that if you have an organism distributed across a wide range, if there's going to be different populations of that organism, and are the selective pressures going to be the same in every one of those populations? No. Okay? As a matter of fact, no 2 populations of even the same organism will have exactly the same selective pressures working on it. Why? Because environmental conditions are different in different places. They just are. Okay? Now, they may not be majorly different, which of course may explain why that organism is there, because that organism may be adapted to a certain set of circumstances and may explain why you find it here and here, but not here. Because the conditions are much more different here than they are here and here. Yes? 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 Okay. All right. So, point is, of course, in any individual population, most of you got this question right, by the way, and I was happy to see it. Evolution or natural selection works on the phenotype, okay? Now, your phenotype, as you know, can be both genetic and environmental. We had this discussion as well. It works on the phenotype to change the allelic frequencies in a population. But selection works on individuals. Selection does not work on the population. Selection works on individuals, which changes what's going on in the population. Yes? Okay. Just want to make sure we're absolutely crystal clear. Selection is working on individuals. Now, it should also be clear, however, that who makes it, who gets their genes into the next generation is not just a simple, this one's better adapted, that one's worse adapted. Because we are, of course, a combination of genes, some of which may be great, some of which may not be so great. Some organism good at finding shelter may not be great at convincing mates to mate with him or her. Yes? So it's the combination of genes that makes you capable of getting your genes into the next generation. Okay? And let me remind you of one more thing. what is best adapted now is not necessarily best adapted later. Okay. As a matter of fact, I think I even mentioned it in chapter 3 that if anything, evolution kind of runs one generation behind. In that the individuals that survive and reproduce now are best at surviving and reproducing in these conditions. Their offspring getting their genes may be facing a slightly different set of conditions. Yes, which means they may not be the best fit. Okay. Now it's not likely to change dramatically, but it can. Okay. And depending on how long, again, your generation time is, it may be quite different. Because conditions facing an insect that comes out 2 months later after the first generation may not be all that different, whereas conditions facing the next generation of elephants, which is 33 years down the road, may be quite different. Yes, and we've kind of made all of these points before. Okay. And the point, of course, is that selection will change genetic frequencies. We did this in lab week before last now. Oh, I'm sorry, we didn't do it at all in lab because we don't have a lab. Got my classes screwed up in my head, okay? But the idea, of course, is that if you add any selection whatsoever, either for or against some genotype, the frequencies will change through time, okay? In the textbook, you remember I started this whole discussion by mentioning they have this hypothetical mouse population with a p-value of 0.6 and a q-value of 0.4. Remember how that I started that? And that's what's at the beginning of this chapter. Okay, partway through their discussion, they add selection to it because if you have, I'm hoping that everybody can do this. If your p-value is 0.6, and that is frequency of big A, and your q-value is 0.4, which is your frequency of little a, everybody's okay, yes? Okay, then you know that your frequency of, and let's make sure we can do this, homozygous dominant individuals in the population is 0.36. Your frequency of homozygous recessive individuals is 0.16. And your frequency of heterozygous individuals is 0.48. Yes. Hopefully you can do this. This is necessary to do your Hardy-Weinberg problems that are due on Wednesday. Let me remind you of that. I am recording. Yes, I did start recording. Yes, okay. Just want to make sure. Okay. Yep. Okay, so we do this, right? Okay, so what the authors did is they hypothetically said, okay, we're going to make it so 75% of these guys survive, but only 50% of these guys survive. So they added selection against the little a allele. Whereas if you have both, then you only have a 50% chance of survival. And if you have one, you have a 75% chance of survival. You with me, everybody? What does that do to the allelic frequencies? What does that do to the allelic frequencies? You should actually be able to do this if I gave you this problem. You should be able to tell me what the new P and Q would be next generation if that selection was applied to this population. Well, the easiest way to do it is to make up a hypothetical population of, say, oh, I don't know, 100 individuals? Because if you had 100 individuals in your population, how many would be this way? 36. So you have 36 big A, and you have 48 big A, little a, and 16 little a, little a. Okay, so if 25% of these went away, 25% of these went away, okay, that would make this become what? All right, let me do the easier one. If half of these went away, that would make this what? 8. Okay, and if 1/4 of those went away, that would make it. Okay, 12 is a quarter, but if you take a 12 away from that, that makes that 36. So this is what they do in the textbook. They simply set it up and say, okay, if you have the little a allele, heterozygous, you have a 75% chance of surviving. You have 100% chance of surviving if you're this one. Now, this is obviously artificial selection. I mean, and I don't mean artificial in the sense that human beings are doing it. I mean artificial in the sense that nothing would actually work quite this way. But it's to make the point, okay? And so if this was the case, then you can do this, because you know how to do this too, because we did problems like this. Everybody understand what I just did? I wrote down the alleles that are in those individuals, right? Yes?
That's like the predicting part.
Yeah, well, no, this is like the adding up part. If you've got 36 of these individuals, it has 72 big A alleles. If you have 36 of these individuals, there's 36 big A alleles and 36 little A alleles. If you have 8 of those, that's 16 of the little A alleles. You get that? Okay, all right. And so this is 160. What's P now? P would be 108 over 160. Somebody do that for me, please. And then frequency of little a is 52 over 160, which I can do in my head because obviously it has to be what remains. Okay. And my point? You add selection, it changes the frequencies. Okay. Yes. What is it? Incredibly artificial mathematical example, but the point is brought home. Is if there's selection against a particular allele, it's going to change the frequencies. Yeah. So might as well do a simple mathematical calculation and show that that is exactly what happens. And it happens exactly the way you would expect. If you add selection against the little a allele, what do you expect the little a allele to do? Go down in frequency, and the big A allele therefore will go up in frequency. Ding, ding. Yes. And that's what they do in the textbook. The reason I was using this example. Okay. Can I erase this? Say something? Okay. Okay. So, not an amazing thing, not an earth-shaking thing, okay? By the way, could strong selection lead to fixation of a particular allele? And perhaps I need to define fixation. I need to define fixation. Fixation means you take the frequency of one allele all the way to 100%, meaning you have lost the other allele. Is it possible to do that? It's possible. Now, is it likely? It's much less likely if the population size is big. Small populations, you are much more likely to lose an allele. And as you're going to find out, you can lose an allele even due to chance in small populations. Okay. But in big populations, let's say little a was 100% lethal, and we're going to actually do this. Say, because there are alleles out there like that. Okay. Let's say little a was 100% lethal. Would it go away from the population? Why not? Yeah, that's exactly it. If you have a recessive lethal allele, will it go down in frequency? By a lot, especially if it starts out with any common frequency whatsoever. But you'll get to a point where all of the recessive lethal alleles are going to be in these individuals, right? And if there ain't many of those, the likelihood of you having any little a individuals in your population are pretty small. Because that would require that one of these mates with another one of these. The only way you would get a little a individual, yes? And even then, when these individuals mate with one another, what's the likelihood of getting a little a offspring? 25%. I'm assuming everybody knows that because you know how to do Punnett squares. Okay? You with me? So the point is that if that individual is running around out there, that's lethal, but it has no effect in the heterozygous individual. It can be maintained in the population. It can be. Doesn't necessarily mean it will be, but it can be. Yes, Sean? Hang on to that. Just let me get to that. Okay, because I'm going to talk about, oh, did you? Okay, go ahead. Sorry. Don't mean to cut you off. Go ahead. Yeah, I'm gonna, like I said, I'm gonna get to that, okay? Because that one's a very interesting one that I want to talk about, okay? All right. And as I have suggested, depending on the environment, you can have certain alleles that become more frequent and certain alleles that become less frequent. And that can be different in different populations, okay? There is a gene in fruit flies that is discussed right here in terms of a nice little cute experiment you can do with selection, okay? What you do is you add ethanol to fruit fly diet. So you end up with a lot of drunk little fruit flies, okay? Now, actually, you don't. Although, to tell you the truth, I have seen the effects of alcohol on certain insects, and they actually can act somewhat drunk. I've seen moths that can't fly because they've drunk some alcohol. Okay. All right. But what's interesting in this case is that there is in fruit flies, let me get the notation correct. Screw this up. There is a gene in fruit flies, PDF. This is an enzyme that helps with normal processing. Let me see, what does it say? Okay. This is an enzyme that is used to process certain bits of food in your environment, okay? And there is this one that happens to be a version of the enzymes that helps them metabolize ethanol, okay? So guess what happened over 50 generations of raising fruit flies on a diet rich in ethanol? They have these two alleles for the enzyme for processing certain food items, and that one helps you with ethanol.
Those.
So they became much more common in the, and that's exactly what happens. Okay, exactly what happens. There's a nice little graph, and this is one of the bigger diagrams they actually have in the book. A lot of times they put these little bitty tiny diagrams over that. They're ridiculously tiny in this book. I mean, it's crazy how small. It's the one really big complaint I have about this book is their pictures. A lot of times they say, here, as you can see in this picture, and he's like, I can't see a damn thing in that picture. What are you talking about? Okay. But this one you can actually see, and this is 50 generations, and these are the control groups. And what you'll notice is in the control groups, the frequency of the S allele hovers around, 20, 30, 40% of the total of all alleles. But in the ethanol groups, it gets up here close to 1, and even does become fixed in one of the populations. Okay, because the populations are relatively small. Okay, yep. So exactly what you would expect. Because it's always nice to say things and have mathematical illustrations, but it's always important to have real examples you can point to. And here's a nice simple one. Okay. All right. Having said that, I was mentioning just a moment ago what happens if you have recessive lethal alleles in the population. And I'm going to go through some interesting examples of various patterns of selection. And I'm going to start off by talking about recessive and dominant lethals. I mentioned it theoretically a moment ago, but told you that I would give you an example. Here, one of those pictures in the side, but you can actually see something in this picture. Cute little beetles that some of you may have actually seen before in your lifetime, okay? Because these are flower beetles, no, not F-L-O-W-E-R, but F-L-O-U-R. If you have an open box or an open bag of flour in a Lazy Susan at home or something, and you open it up after not having made anything for half a year, and you see these little black and brown things crawling around in there, they're very likely to be these little beetles. Because it turns out they do very well on ground up grain. Funny how that works. They like flour, okay? Well, it turns out that there is a recessive lethal allele. Now, it's not common in most populations, but you can experimentally manipulate your populations if you can genetically determine what they are so that there's a higher representation than you would normally get. Because what you do is you go, well, I've genetically identified these individuals as heterozygotes. And what you do is you just grab a whole population of heterozygotes. And if you grab a whole population of heterozygotes, that means you are 50-50. Yes? 50 big A, 50 little a. Okay? Well, this little diagram in the middle, and again, it's another reasonably nice one because it's actually in the middle of the text and not one of these little graphs over here on the side. What this shows you is they started from 50-50, and very quickly, the line, by the way, is the mathematical prediction. The line, okay? And this line, of course, is what you expect to happen to the non-lethal allele, the dominant one, okay? And so the mathematical model suggests it should get very low and it should drop really quickly to begin with, but it should get to a point where it should more or less even out. Gets infrequent or rare enough that heterozygotes rarely find one another, and it persists in low frequency like we were just discussing, okay? Well, what the dots are and the little triangles are along the line are the actual results from real populations, where they started with a whole population of heterozygotes, and what happened to the recessive lethal? It dropped off. And what's really nice about this particular example is mathematical models are useful only to the point where the mathematical models are predictive. I'm actually not a big fan of a lot of mathematical models because a lot of biological circumstances, I think, are too difficult to model mathematically and get them right. But in this case, it was just feed them flour, let's see what happens to this allele. That was it. And what's really nice is that the model almost exactly matches what actually happened, okay? All right, you get to a low number, but it doesn't necessarily go away, okay? Now, you should be looking at what I wrote on the board and thinking, okay, I don't even understand why he wrote the word dominant lethal on the board. You should be thinking, dominant lethal. That means anybody who has the dominant allele dies. That would be anybody who's big A, or big A, little a. So anybody who has it is dead. How does that even work? And that's a very valid question, because obviously there are some alleles that if they get mutated would be that dramatic. Every individual with that allele would die. And they never even developed probably, so you would not even know they existed. You got me? However, there are some dominant lethal alleles out there that do persist in the population. And I know you're all thinking, how the bleep does that even work? And the answer is this. How many people have ever heard of a disease called Huntington's chorea? Does that sound familiar to anybody, Huntington's chorea? Okay, Huntington's disease? Okay, it is controlled by a dominant allele, it is always lethal, but it is not lethal until later in life. Ding, ding. This is how a dominant lethal allele actually persists in a population, because it doesn't affect the individual until after they've had a chance to reproduce. It's like around 40. That is exactly right. Huntington's disease around 40 starts to affect you, and they decline quickly after that and die. Kills everyone that has it. Okay? But because it does not affect the individual till later in life, it is an allele that can be passed on. Okay? Now, having said that, even so, is it more likely for that allele to disappear than the recessive allele? Probably so. Again, because not every individual is going to reproduce. For that matter, if you know you have the possibility of having it, you often will get genetic testing done. And if you find out, yes, I have it, you may not have children. Okay. Now, in nature, if there are such alleles, and we assume there probably are for other organisms, obviously both can persist. And my question is probably meaningless because it depends on what time of their life they die, how many offspring they could have, those kinds of things. Yes? 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 Okay. Did you have another question? So. One of the nice things about living in today's world is if we know what alleles we're looking for, we can do genetic testing to find them. Okay. Now, we don't know all alleles for all genes yet. We may at some point, because we now have done the entire genome of several human beings. Okay. And where having gone through 4.3 billion base pairs, which is what a human being is, was unthinkable 30 years ago. We've, like I said, now done it, and it can be done in, I want to say days now, if not weeks. And so it's the kind of thing that, what was once unthinkable is now not commonplace, but perfectly doable. So yeah, if we know what allele we're looking for and we know where it is on a given chromosome, we can go and look at it and find out. So we do have that ability now. So it's nice that we can do that for people who might be experiencing that. I mean, you've heard of this before, because people who have, we know all about cancer not being a good thing, and we know all about people who have certain genes that give you a propensity for a particular kind of cancer. Okay, and you test for those. Okay, if you have one of those genes, you are persistent about getting examinations done and taking care of yourself and doing all the things you would normally do if you think that you might get this kind of cancer. So anyway, so we good with that? Again, I don't know that that's a question you can answer, because different dominant lethals will have different effects at different points in life. And, if it doesn't kill you till you're 60, probably doesn't have much of an effect at all in a human being. If it kills you when you're 22, you might have offspring, but you also aren't likely to have many. And anyway, so I don't know that you can say which is stronger. It depends very much on when and how bad when it expresses itself and how bad the dominant allele is. Okay. All right. Okay. Now we're going to look at something slightly different, and that is selection on homozygotes versus heterozygotes. Okay, and this is why I was telling Sean, just wait a second, because here's one of the, I'm going to even use his example. Okay. as you may have noticed, you can use anything to designate alleles. Any symbol you want. Okay, it could be 3 letters, it could be 8 letters. It'd be kind of stupid to use 8 letters because you might as well just name it at that point. Okay, but what's HB stand for? Anybody? Yeah, very good, very good. I was hoping somebody would come up with that. And how come it's not HG? Because hemoglobin, you would think HG, not HB. Why is it not HG?
Latin, I guess. Oh, periodic table, bro.
What about it? Look at element number 80. What is it? Oh my God, somebody knows. So somebody better know what.
Mercury?
Yes, mercury. HG is in our lexicon as mercury. You would never use HG here because everybody would be going mercury. Okay, I'm just pointing it out to you. Okay, not that it's something you have to remember, but that is why that is HB and not HG. Okay. Anyway, this person is normal. You know how this works. This person is this person, I made it a sickle. What do you know? This person is sickle. Okay. Now, they are this again, capital H, capital N, capital S. I've tried to make it so that it's very clear that this is a codominant circumstance. Okay. If all your cells are sickle, you're dead. If all your cells are sickle, you're dead. Okay. I don't know if blood transfusions can save you or not. I'm not a scholar of sickle cell anemia. Okay. I do know, however, that heterozygous individuals, half their cells look normal and half their cells look sickle. Okay. Now, this does reduce the oxygen carrying capacity of your blood. It does, okay, which means that in circumstances like this, sitting around the table, we're pretty much fine. But if we did a lot of hard work, that someone who is normal would have an advantage over someone who is sickle. Yes? But there are certain places in the world where that heterozygote would be selected for. And those places in the world would be where, well, why? It's because the malarial parasite is a blood parasite. It actually gets inside red blood cells. Guess what the malarial parasite cannot get inside of? Is sickle cell red blood cells. And it's hard for them to get from cell to cell and reproduce and multiply in a sickle cell individual, which means a sickle cell individual is resistant to malaria, which means that at least in some parts of the world, this genotype may actually be favored under certain circumstances. Okay? And so here we have a circumstance where the heterozygote may be favored. Okay, now not always, obviously. Okay, do we have a name for selective advantage for heterozygotes? The answer is yes, we do. It is called overdominance. I'm going to get what I want. All right, so overdominance is when you have selection for heterozygous individuals in a population. Yes? And again, it may be different from place to place, as I was just saying. Because clearly, being sickle, being the heterozygous individual, is not selectively advantageous everywhere. It just may be in areas in the world where there's high malarial occurrence. Okay? So overdominance is selection for heterozygotes. What do you suppose selection for homozygotes is? It would be underdominance. Is selection for homozygotes. Now, I'm going to give you an example that they also give in the textbook. This is the example that is on pages . The main diagram is on page 210. Okay. And what we have is Drosophila with what are called compound chromosomes. Now, what the heck is a compound chromosome? Good. I'm going to do it this way. Okay, if I draw that on the board, I'm going to assume you know what I'm trying to represent. I'm trying to represent a chromosome, okay? A chromosome that has two chromatids. Thank you very much. Again, I'm hoping that's all terminology everybody can handle. Two chromatids, a chromosome with two chromatids. We know that during mitosis, you will separate at the centromere and pull these two separate poles. Yes? Okay. We good with that? All right. So this is one arm and this is the other arm of the same chromatid. Well, what a compound chromatid is when it separates at the centromere, but instead of separating the correct way, it separates this way. So instead of getting an entire chromatid going that way and an entire chromatid going that way, you can get both chromatids or both of one arm of the chromatid going one way and both of the other arm of the chromatid going the other way. Yes? So it doesn't split the right way. Yes? Does not split the right way. Okay. So what's the end result of this? Well, understand that the genetic machinery during mitosis doesn't know exactly what to do with these. Yes? Because it's not right. It's not correct. Okay? And what can happen is that you can end up with gametes that get both of those. You can end up with gametes that get one or the other of those. Or you can end up with gametes that get neither of them. Yes?
You said mitosis.
Mitosis. But understand that if you do that with mitosis, that you're also going to have the same thing with meiosis. Okay? Because it's during mitosis that happens. But then if that happens in the stem cells for making gametes, these are the possible gametes you can end up with. Yep. You with me? All right. Now, point to make. What are the gametes you could get from a normal individual? The gametes you could get from a normal individual would all be that. Yes? All of the gametes you get from a normal individual will be that way. Yes? And so if you made a Punnett square here, I know this is not your typical Punnett square, but would any of these be viable? Well, what do you end up with here? What you end up with here are, in essence, 3 copies of all of the genetic information, right? Because you end up with that, or that, or we get better at this, or that. That's what you could potentially get if someone with a normal chromosome mated with somebody with a compound chromosome. Would any of those survive? None of them would survive. That one's triploid. That one's got 3 copies of one arm and 1 copy of the other arm. That one's the same way, 3 copies of the other arm, 1 copy, and that's haploid. You don't have a diploid individual. None of these are viable. Yes? You with me? So some Drosophila with a compound chromosome, a chromosome that didn't divide properly, mates with a normal individual, what's the survivorship of their offspring? 0. Okay? How about, however, if they mate with another individual with compound chromosomes? So they mate with another individual who is got gametes that look like this, gametes that look like this, gametes that look like this, and gametes that look like that. Do any of them end up being viable? Because remember, to be viable, you would have to have the normal diploid complement. Normal diploid complement. Would that have the normal diploid complement? That would be quadruploid. I'm sorry, tetraploid. Yes? That would have 4 black arms and 2 red arms. That would have 4 red arms and 2 black arms. Yes? What about this one? If that gamete fertilizes this gamete, you end up with the appropriate amount of genetic material in it. Yeah, it's diploid. Okay, it's got 2 black arms and it's got 2 red arms. Yes? Yep. Okay. So this one doesn't work, this one doesn't work, this one doesn't work, but that one, check mark. It's fine. Yes. Yes. Was that a C?
Yeah.
My point? Well, it ain't great, but if you have 2 individuals with the same compound chromosome, homozygous or compound chromosome structure, weird. 2 individuals, same compound chromosome, you have 25% survivorship in your offspring. 25%, 4 out of 16. Okay. Whereas if they cross with a normal individual, you have 0 survivorship. So only when you cross with someone of the same genotype, the same homozygosity for that compound chromosome, do you get viable individuals. Like I said, it ain't great. It's a weird example, you must admit, because even those that have the same compound chromosome, survivorship is still only 25%. But 25% is, needless to say, better than 0. Yes? So, like I said, weird example, but I kind of like it, okay? Because it really does show you try to mate these with something different and they ain't going to produce nothing viable. They can only mate with individuals with the same genotype to get viable offspring. Okay. Yes. You with me? Okay. So there's that. That's an example of underdominance. And then this is one that does not get enough attention. I really like it a lot. That is something called frequency-dependent selection. Now, I think you will notice that what we're talking about right now are some really interesting special cases. Okay. What I haven't talked about yet are directional stabilizing and disruptive, which you've probably heard about multiple times in multiple classes. And I will get to that. It's just that we're talking about these really weird special patterns of selection right now. Something called frequency-dependent selection. Okay. Now, this is well named because frequency-dependent selection is selection dependent on frequency of individuals. And again, I'll use an example from the textbook. I'll show you the diagram. This one actually is also kind of okay. Okay. It's this one on the top of page 213. And it shows elderflower orchids. And I think you will notice that the elderflower orchids, there are these kind of pinky lavendery ones and there's these yellowish whitish ones. Well, they're purple and yellow is what they're called. Okay. All right. Now, yellow is recessive. Purple is dominant. And if there is just, if there was random mating and equal numbers of yellow and purple alleles in the population, you would expect about 75%. I got it backwards. Yellow's dominant, purple's recessive. Okay. You would expect about 3/4 yellow and 1/4 purple if everything was completely even. Yes. Yep. Okay. What's interesting is you kind of get that, only it's 77% yellow and 23% purple is where the population ends up at every single time. Now, the question is, why does it always end up there? And when I say end up there, I mean this. You establish a population in a little field, because this has been done, little population, okay? And you let the pollinators just go. If you start with 0.6 yellow and 0.4 purple, it ends up at 0.77 yellow and 0.23 purple, which means the pollinators are selecting for that level, okay? If you start at 80% yellow or 85% yellow and 15% purple, it'll go the other way to 77% yellow and 23% purple, okay? Which means if yellow is more frequent, it will become less frequent. If yellow is less frequent, it will become more frequent. Its abundance depends on its own frequencies. How many yellow, how many purple? And the pollinators pick. Now, why does this happen? Well, what's really interesting is there's a little sort of side story in this story that these orchids do not produce nectar. And the fact that they don't produce nectar would suggest to you that the bees shouldn't visit them at all. And the picture does show you a field that's full of these orchids. And if that was the only flower in that field, how many visits would pollinators give these flowers? The answer is 0. Okay, if that was the only flowers in the field. But they're not the only flowers in the field. And so they're buzzing around. Why do you suppose yellow orchids are preferred over purple ones, even though neither give them any nectar? Okay, what about the color? Okay, but there's something else going on as well. And that is the other flowers in the field, there's a lot of, that's it, is that in the field, there's a lot of other yellow flowers that offer nectar, okay? Now, the question is, if that's the case, then why are there any purple elderflower orchids at all? It's a really good question, and there's a really good answer. Because if yellows are visited more frequently, and it's because there's other yellow flowers around that have nectar, you think, just throw a bunch of yellow flowers out there and you'll be fine. You'll get pollinated because the pollinators will come and go, well, that one didn't have any nectar, but I'm going to the next one. Well, there's a really good reason why having purple out there. Because what happens is, if it's solid yellow, because this has been done as well, if they've planted fields of solid yellow and solid purple, all homozygous, so you can't get the other one. Neither of them is visited as much. So why have two colors? It's because if you go to a yellow one and go, oh, I didn't get any nectar there, and there's a purple one next to you, it's like, oh, different flower, another shot. We'll try this one. It's about variety. It's about you're providing the pollinators with alternative targets. Maybe, just maybe, this time when I go to the purple one, there'll be nectar. Oh, no nectar. I'll go back to the yellow one. And so it gives them multiple targets as opposed to just one. And obviously, having 23% purple is the ideal for the pollinators. Okay. You with me, everybody? It's a really fun little story in that neither offers nectar, and yet there's two different morphs. The yellow one's more common. Why? Because there's lots of other yellow flowers out there with nectar. But the purple one is there to keep the pollinators interested. You with me, everybody? Okay. What? Yes. Yeah. It's in my eyes, that's not purple at all, but they call them purple. Okay. All right. I'll give you another example. This one's fun too. Everybody recognizes this noise. What noise is that? How about a mosquito, like next to your ear in the summer? Like, go away. Okay. Who's making that noise? I know it's a mosquito, but I'm asking for a more specific answer. Who's making that noise? Okay, that's okay. That's what's making the noise, and who's making the noise is male. Okay. What's making the noise actually is the wings. They're flapping so fast that they actually make a tone. Okay. Now, why do they do that? Why do males do that? Why do you suppose they evolve so that their wings actually make a noise? Because it attracts the females. This is why. It is their way of singing. They don't have any vocal cords, but they can sing. Okay. Well, in some species of mosquitoes, if females hear, and then the next one they hear, the next one they hear, and then the next one they hear is, you know who they mate with? The different one. Okay. It is the one that is less common. Is that frequency dependent selection? Exactly what it is. The ones that are less common are ones that get mated with more. And what does that do for the population? What does that do for the population? I'll answer your question in just a second, but answer my question first. What does that do for the population? Increases. It increases the uncommon ones, which means you're maintaining variation. Because as soon as the uncommon one becomes more common, then what happens? They become common, which means now the females will select a different one, and then select a different one, and then select a different one. They keep selecting the rarer or more novel ones, which helps maintain variation in the population. In this case, you do. You get waves of selection, if you will, in that you get less common to more common, then another less common to more common. And the point is, eventually you'll come right back around to this one again, which was more common for a while, but now that it's more common, it's not being selected for, and so it'll drop off and it'll be selected for again. So if you're talking about waves being like that, sure. Now, that's for frequency dependent selection. Understand, this is not how all selection works. I'm giving you some interesting examples. Yes? Okay. Now I'm going to show you a picture.
Why did they,
What?
why did they go?
Probably because novel means potentially something. I need to qualify this. Why do they do that? Because something in the novel song means better fit to the female. What means better fit? We don't know. But something in having a novel song, because understand, if the female doesn't think that that's going to provide her with good offspring, she wouldn't do it. Okay. Now, of course, we all understand that she's not sitting there thinking about it. It's instinct. Okay. But still, obviously something has selected for them to pick novel males, because novel males for some reason mean better fits. Okay. All right. I know that's a really fun one. That crap.
Okay.
That's what I was going to say. Okay. Okay. Told you I was going to say that. Oops, need this. I'm going to give you one more example. Now, I don't know that this is actually called the European forest snail. I'm sure I knew the name at one point, but at one point I had forgotten the name, and so I just started calling up European forest snail images, and I get what I want, and so I never relearned the actual name. So, but you will see what I mean when I talk about the variety of snail shell patterns in European forest snails. Some are brown, some are gray, some are yellow, some are orange, some are striped, some are more boldly striped. And you see all these images, not just the main one that I have on the screen right now. Let me turn this way, okay? But the ones across the bottom as well, okay? Yes? Now, why are they these colors? Why brown, why gray, why orange, why yellow? What about the environment? I said forest. Where are they? On the ground. What's on the ground with them? Leaves and rocks. You're not wrong. Ground, brown, rocks, gray, okay? Although it doesn't show really well here, there are solid gray, there are solid brown, okay? Yes? Okay? All right. These guys also enjoy frequency dependent, well, okay, enjoy is not the right word, but experience frequency dependent selection all the time, okay? And it's because of this. All these different colors will blend in one way or another with what's on the forest floor, okay? Are they pieces of food? Absolutely. Birds are hopping around the forest floor looking for these guys. Who are they going to eat the most of? The ones they can see. Who are they going to see? The ones that are most abundant. Those are the ones they're going to see most easily because they form what's called a search image. They form what's called a search image for them. They find one. If there's another one that looks like that one, suddenly they go, oh, that's what they look like. And now it's easier for them to pick it out from the background than the other ones. Why? Because they've seen it a bunch of times. And of course, the end result of that is exactly what we were talking about, only in the other direction with the mosquitoes. In this case, The ones that are more frequent are the ones that are selected against. Well, actually, it's the same as the mosquitoes. The ones that are more frequent are the ones that are selected against, and those that are less frequent are not seen, even though they're out there, because the birds start focusing in on this one image. And of course, after a while, the one they focus in on becomes less common, and others become more common, and they'll switch. Yes? Great example of frequency-dependent selection. So there, I gave you 3 examples of frequency-dependent selection: the elderflower orchids, mosquito mating song, and these snails. Okay? All right? So kind of fun, weird patterns of selection. Okay? And again, one point I want to make about frequency-dependent selection is that frequency-dependent selection is really useful in an evolutionary sense in that it maintains variation. It maintains variation. Because those that are less common become more common. Those that are more common become less common. None of them go extinct, however. They all are maintained in the population. Yes, maintains variation in the population. Okay. So we good with that? Like I said, these are some weird patterns of evolution. We will come to the more common patterns, namely directional and stabilizing and disruptive, later. Okay. I told you that the rest of this chapter, however, was talking about the first 2 assumptions of Hardy-Weinberg. One was selection, and we've now had a little discussion on various patterns of selection, and the second one was mutation. Okay, now there's not a lot to say about mutation that you don't already know. Actually, one, mutation tends to be good, bad, indifferent. Bad or indifferent, okay? And of course, since it tends to be bad, that means it ultimately is selected against, and I'm going to come right back to that here in 3 minutes, okay? It should also be clear, however, that mutation, in order to affect a population, has to take place in cells leading to the gametes. I made a point that, how many people have ever seen somebody with a shock of white hair? Okay, that's a mutation. There's a mutation that occurred in cells leading to the growth of these hair where melanin is no longer produced. And does that affect this person's offspring? No, that is a somatic mutation. Can I say that? Somatic mutation? People have heard of somatic mutations before. You know what I mean when I say somatic mutation. Time for me to define it? Okay, what's soma? What's soma? What's some, as in chromosome or ribosome, or what's some? Lysosome. Some. What? Not a cell. Remember, site is cell. Something simpler than that. Chromosome. Chromosomes, that's a really weird name for what they are. And you'll see why in just a second. Because you know what chromo means? Wow, we all need to have some more education on our Latin and Greek roots. Okay, some, or soma, means body. Chromo means, I know this is sort of an aside, but chromo means colored. Chromosomes were named colored bodies because the first time they looked at them with a large enough microscope and stained them, they were colored bodies. They had no idea what they did. They just simply called them colored bodies.
Is that just with the bright?
That's with staining them well with some stain, and that's the color. Like I said, really weird. It had nothing to do with their function at all. And it's because they had no clue what they were when they first saw them. You get it? Okay. But somatic mutation is a mutation that occurs in a cell of the body, a skin cell. Okay. Why does that matter? Because that's never getting passed on to offspring. Never getting passed on to offspring. Somatic mutations. Understand, we all have, I've hopefully made this point already. We all have somatic mutations. We all do. Okay? Because mutation is a normal part of cell division. Normal part of cell division. We're all mutants. I've said that now like 3 or 4 times. Yes? Okay. But the somatic mutations don't matter in evolution. They don't, because they only affect that individual. They don't affect offspring or anything else. The mutations must take place in germ cells, in stem cells leading to gametes. Yes? Or somehow get passed to stem cells leading to gametes. Yes? Yes? Okay, so that's one thing. How frequently do those mutations in those gametes get used? Not very often. We all know that it's a crapshoot. That's probably a bad way of putting it, but it's a crapshoot which gamete fertilizes which gamete. Yes? Okay. So, is mutation by itself going to change genetic frequencies quickly? Mutation by itself? No. Mutation is very slow at, by itself, changing genetic frequencies. Very slow. Snail's pace. Okay. And as we said, if it's a bad mutation, it just gets eliminated. Yes? So mutation is not going to change frequencies by itself quickly at all. Okay? Now, can you have a mutation, by the way, could you have a mutation that changes a dominant allele to recessive or recessive to dominant? Does that happen ever? Is that possible? Sure, if there's one base difference between them, that base could mutate. You could get a dominant. Would that change allelic frequency? Sure. How fast does that happen? Not fast at all either. Okay, so it doesn't matter what the mutations are occurring in. It's not going to change allelic frequencies by itself quickly at all. But I will point out one other thing, and that is that although mutation cannot change allelic frequencies quickly, in the end, mutation is everything. It's everything. Because understand that in the long run, the only place where new alleles come from is mutation. Now, I could qualify that, however, by saying, of course, new alleles can come from, what am I looking at here? I've got a minute. New alleles could come from some other organism, virus injecting DNA, or something called lateral gene transfer, which we'll talk about later. Okay? Yes? So mutation doesn't change anything quickly in the short term, but in the long term, mutation is everything. Especially when you combine it with who? What's the first thing we talked about today? Patterns of selection. When you combine mutation with selection, that's when it becomes potent. Now, it should be clear, I'm going to say 2 more things. It should be clear that most of the time there's going to be what's called a selection mutation balance, where since mutation is more frequently bad than good, selection will simply remove it. That's what's called the selection mutation balance. You with me? But because mutations are not always bad, so because mutations are good, selection can run with a good mutation and make changes much more faster, much more quickly than mutation could by itself. Yes? And that's where we will start up next time.